This is the Soil Health Assessment Center, and we are now in the College of Agriculture, Food, and Natural Resources at the University of Missouri. And this is just a listing of some of the analyses that we do here at the Assessment Center and some of the packages that we have. So you can see we do potentially mineralizable nitrogen, total nitrogen, active carbon, total organic carbon, water-stable aggregates, pH, salt and water, effective cation exchange capacity, exchangeable cations, and plant-available phosphorus. In addition, bulk density, phospholipid fatty acids is the analysis we use for our most extensive microbial analysis. We also do neutralizable acidity, particle size, and other analyses that the lab did as a characterization lab that it was before we switched to or added soil health to our analyses. I think that manure will affect nearly all of these analyses, as Bianca was saying, and most of them in a positive direction. So one of the questions we had was, when do you sample? You can sample in spring, fall. January may be best for potentially mineralizable nitrogen. Sample when you have an appropriate moisture content, so the soil's not saturated. Most important is be consistent, because the analyses, the results you get will vary upon when you take the analyses. And to be most consistent, take it at the same time of the year. And then every 3, 4, or 5 years, depending on the rates of change or in your management or crop yield improvements. This is a page from our recent reports that we've developed. One of the problems or concerns that Bianca was talking about was nutrient availability or imbalances when you have manure added. This is an example of phosphorus. This is some of the results, just an example that we use for training and showing that you can get in the samples across the state just a wide variety of concentrations of, say, phosphorus. The same would be found with mineralizable nitrogen or total nitrogen. We have been able to gather a lot of information over the past year because the Department of Natural Resources in Missouri has issued a cover crop cost share program, and as a part of this, they have required the farmers who are participants to send a soil sample to our laboratory, and they did this so that we could build up a database for soil health properties within the state. So we had samples submitted by about 73 out of the 115 or 14 counties in the state, and this graph will show you how many samples within a range that we received from which counties. So we received samples from most of the state except for the south central region, which is the Ozarks, where there's very little cropping. Now this will show you an idea of why we did this. As Bianca's and everybody else, a lot of other people would know that when you have a measurement, you need to know what kind of soil you're talking about. So here we've shown different regions in the state showing the average that we got for total organic carbon. So if you see, if we have a sample submitted from the Boot Heel region with 1.4% organic carbon, that's average. But if we had it received from the Northwest region of the state, that would be well below average. So you can see the range we have across the state, it can range 50% higher than what it is at the lower part. We are starting to get more information about how manure affects these properties, and what we have here on the left is just a list of the analyses that we ran on the samples that were submitted from the cover crop program, and on the right there is a questionnaire that people submit. So they have personal information, they list what the landscape position was of the sample, they list the soil, and crop rotation over the past 5 years, tillage over the past 5 years, what they plan to do for the next 5 years, and of this season, we have added some manure questions about the manure added over the past 5 years, what type it was, how many tons per acre, and how it was applied. Another source of information for the effects of manure on soil health is Sanborn Field. Sanborn Field has been a continuous cropping system from 1888, so it's 127 years. There are different rotations. Some crops have manures, or some plots have manures added. Some of them have different fertilizer regimes added. Some have cover crops. The one disadvantage is most of the treatments have not been replicated. But in addition to that, we will be able to build on yield data that's been collected. There's a weather station there. We can compare to research conducted through all these years. And it's also a good place to take people, and people can see, feel, touch the differences in the soil over a short distance. Then the example of the data from Sanborn Field, here you can see we have 3 different cropping systems. We have continuous corn, continuous wheat, and a rotation with corn, wheat, and red clover. So with no fertility, this is water-stable aggregates. You can see that the corn was well below the wheat and the rotation. Then you have full fertility with fertilizers added, and the corn has been boosted, but the other 2 are still well above it. But if you add 6 tons of manure, it greatly affects the corn system, but not to the same extent as the wheat and the more extensive rotation. So this is the sort of information that we can gather through this, these plots and the ongoing research there. And we have a graduate student, Saranya, who's working with Dr. Randy Miles to establish what we have going on in these different plots. We also have applied and received a manure and soil health mini grant, where we're going to analyze the data from Sanborn Field, current and in the past, and then compare the cover crop samples where manure has been added, what kinds of manure has been added, the different rates to the soil health properties that were from the other samples in that state in the same region. And eventually we would like to put the samples categorized as to major land resource areas, so we can tie it back even more closely to the soils. And that's where we are right now. We will hope to have a lot more information as we have analyzed the samples and made the comparisons.